Hi and welcome everyone. My name is Shoaib Iqbal. I'm an assistant professor at the Department of Pharmaceutical Sciences, Feik School of Pharmacy. It is a pleasure to present my talk today on polymeric nanocarriers as a delivery vehicle for RNA. I would like to share my goals for the talk. In this talk, I'll be going over some of the rationale behind these genetic drugs and some of the barriers that they encounter as we try to use them as drugs, as well as the motivation behind nanocarriers. And I'll share a couple of stories of polymeric nanocarriers as a delivery vehicle for these RNA therapeutics. I find it quite useful to give the forecast of the talk so that way the audience can follow along as we move during the talk. So, as you can see on this slide, I like to show this slide to emphasize the point that, or make this statement that it is very difficult to develop new drugs. And this is a chart that describes the new FDA approval over the past 20 or so years. We can see that the one presented in blue are some of these new molecular entities or the small molecules, while the one presented in red are the biologics. We can see that these are the outcomes of all the global efforts that have been performed over the years or in each respective year to make these drugs into the approval or to make these drugs into the clinics. We can see that these are the outcomes of global efforts, and these are done for the small molecules. Hundreds of thousands of molecules were tested before we were able to get a handful of approximately 30 on average each year that got into the clinic. For biologics, the odds are more or less the same. We can see only a dozen or so molecules, new molecules that get approval into the clinics. And this is all after the global efforts and after all those efforts and hard work of the scientist. Many times we can see several case studies that even post-approval, some drugs are withdrawn from the market because of some of the adverse drug effects. So this emphasizes the points that development of new drugs is a challenging process, and it requires a lot of hard work and effort with success rate that are on the very low end. I also like to show this in this slide, the basic high school biology. We all know the central dogma where we have the DNA transcribed into an mRNA, and mRNA is then going to translate it into the protein. All of our proteins are encoded into the DNA, and if we can control the genes, we can control the expression of the protein. Many of the functions within the cells are performed by proteins, as you can see in the center of this graph, in this picture, that the protein, in case of disease state, we could either have an upregulation or downregulation or a misfolding of the protein. And when one of these three instances occurs, we could see the progression of the disease or the onset of disease in its function. But when we look at the traditional drugs, the one that I showed you on the previous slide, in case of small molecules, they have to bind that specific site onto the protein to correct the function of the protein, or the ones for the case of biologic on the bottom. We can say that those antibodies, most of the time, they have to they have to bind with the proteins or bind with the receptors, antigen antibody interactions for them to exhibit their therapeutic response. So this is the traditional way of treating the disease using conventional drugs, whether it be small molecule or the biologic. However, if we look at this central dogma, if there is a way in which we can either prevent the translation process to occur at first place, we can prevent the protein, the disease-causing protein, to express in its first place. So this is going to be a more potent and it's going to be a more significant response because we have prevented the expression of the protein, the disease-causing protein, at first place. Or sometimes, as there are some genetic drugs, or we know there are processes where we can interact at the DNA level, and even before transcription could even occur, we could prevent the progression or the process of production of protein. And in those cases as well, we can see that we can prevent the disease-causing protein to express within the cells. And this is advantageous compared with the traditional drugs because in case of traditional, we have the protein that is being expressed, either upregulated or downregulated, and we are just trying to minimize the impact of the protein by using small molecule or biologics. In case of the genetic drugs, we can, or if we just prevent or tackle the problem at the transcription or translational stages, we can improve the efficiency of the therapeutic response. This slide shows the rationale behind genetic drugs. We can see that we have a traditional small molecule. These are usually on the smaller scale in terms of their size, less than 1,000 Dalton. On the right is a more informational drug or a genetic drug, and this example being shown here is of an siRNA, but it is applicable to all other variations of RNA, like mRNA or microRNA or antisense oligonucleotides. We can see that in terms of size, siRNA is a big molecule. It is 30 times larger than the small molecule drug, and they are usually going to have a coded and as well as a non-coding sense and antisense strands in case of RNA. In case of properties, for small molecules, we can see they could either be a hydrophilic or a hydrophobic molecule. But in case of these RNA drugs, these are usually on the hydrophilic and water-soluble molecules. And we can see in case of small molecule drugs, we can have permeability dependent largely on the properties of the drugs. With some of them, hydrophobic ones are very permeable. But in case of these RNA informational drugs, they have very poor membrane permeability, and this is largely the result of the size as well as the negative phosphate groups that are part of these nucleic acids that prevent their easy access within the cells. On the right, I have tried to show some of the targets that we have at our disposal to develop new drugs. In case of new molecular entities or these small molecules, the available targets is a small subset. So we have to have a very targeted and a very serendipitous approach for them to discover and develop new drugs. So we have a handful of targets that we could target and develop the new small molecules. The odds are better in case of biologics because of the receptor binding, because of the antigen antibody response and interactions. So in case of biologics, we have a better targeted approach in terms of the development processes. So we have slightly better odds of developing new drugs. But in case of informational drugs, since we know that if we have the complementary sequence, we can target any gene. So in case of these informational drugs or these genetic drugs, we can target any specific gene of interest, and we can, since these nucleotides have more or less the same chemical properties, so we can potentially target any specific gene or any specific protein and use them as a potential drug candidates. While I have described some of the useful potentials of these genetic drugs, the problem and after having a discussion on all the good things that we could achieve with these things, with these drug candidates, they have some inherent vulnerabilities, and this has largely due to the nature of these genetic molecules. If we try to administer them into the body, inject it within the body, we will see that these are easily cleaved by the serum nucleases that are present within the blood. They are very small molecules, so they are going to be readily filtered by the kidneys. They are not going to accumulate specifically, so they have a non-specific accumulation, and their anionic charge, highly negative phosphate groups, they offer a highly negative surface charge to these nucleic acids, and they are very hard to be taken up by the cells. However, based on the preclinical studies and preclinical work that has been published to date, we have started to see quite early on that they can specifically silence the gene of interest once we administer them within the body. And in the animal models, it was shown that siRNA, as shown here on the right, was able to silence, or that siRNA target gene was silenced by the siRNA. And in its unmodified version, or even by conjugating them with cholesterol, it was able to exhibit its or perform its therapeutic function. So these are they have the great developmental potential to be new drugs. But if we just address these delivery hurdles or these barriers that they encounter, and we just try to address them, we could achieve their therapeutic potential, and it is going to open a realm of therapeutic opportunities for treating various diseases. And in my examples, later on, I'll show you a few case studies of how we could achieve these potentials. For any of these genetic molecules, nucleic acid drugs, RNA drugs, the biggest delivery hurdle is of passing through those cellular barriers. We can see that these are highly anionic molecules, and their size is pretty large. Getting them into the cell is going to be the first challenge, and even after the cells, we know that whether it is targeting transcription or translation, it has to get into the certain compartments within the cell. So having it escape the endocytosis or the endosomes are going to be the key challenges, and this is some of the stuff that is usually tackled by using a delivery approach. And we can see that in case of a delivery is actually is a combination of firstly the cellular uptake as well as the endosomal escape. Only the RNA drugs are going to be delivered once they are taken up by the cell as well as they escape the endosome. While when we use the general term of uptake, it means only a process of entrapment or endocytosis of these nucleic acids. And we have quantified, or various studies have quantified the amount of the drug that is able to escape through these nanocarriers. And it's going to be usually less than 1% of the total drug. So this indicates that the therapeutic potential that these nucleic acids are very potent molecules, and they can once within the cells, they can easily we just need to have an escape potential of nearly 1% or less than 1 for them to exhibit their therapeutic responses. And this slide outlines a very simplified view of approaching our delivery technology. We start with the therapeutic goals as well as the cargoes that we are trying to deliver or use. In that delivery system, we draw from our experiences or from the experience of others in the field, and then we incorporate some distinct features and we deliver. And when we say deliver, both the uptake as well as the endosomal escape. And the ideal or a good way to approach this is to incorporate both formulation practices as well as chemistry and biology approaches to address all the delivery barriers, challenges, and or to tackle over the delivery hurdles along the way. So in my first story, I'm going to share an example of a polymeric nanocarrier as an RNA therapeutic. So this was more like a polyester-based nanocarrier. We used PEG PLGA along with the cationic lipid. That is a very traditional way of delivering these nucleic acids. So this was more like a hybrid polymer lipid nanoparticle, and we used siRNA. And upon using formulation science, using double emulsification, we were able to obtain some nanocarriers. But the main point behind, or the idea behind this work was we have seen a lot of cationic nanoparticles that are being used to deliver these nucleic acids. If we could tackle, or if we could tweak the size, tweak the surface charge of these nanocarriers. So to address or to achieve that, we were able to use a lipid that offered multiple amine groups, primary as well as a secondary amine, that we could then carboxylate to tweak its surface charge. And this allowed us to have a range of surface charges ranging from highly cationic, that we named as aminated nanoparticles, to plain nanoparticles having more or less nearly neutral surface charges, or carboxylated nanoparticles which were on a relatively negative end. And by just modifying the surfaces of these nanoparticles, we were able to get a library of 5 different nanoparticles, and by just tweaking the level of carboxylation, we were able to achieve these different surfaces on these nanocarriers. And to test them to see if having a different surface charge will impact the therapeutic outcome of these nanocarriers. We were able to characterize these nanoparticles and were able to validate that there was a different surface charge on these nanocarriers. We were able to see more or nearly constant loading of the cargoes within these nanoparticles, and the diameters were also not impacted during the process of charge conversion. We were able to see that these nanoparticles were quite stable, and we used the phenomena of FRET, and we determined the FRET efficiencies, and we were able to see that these nanoparticles were intact in more biological milieu, while in case of organic phases, these nanoparticles were no longer able to withhold themselves, and they disintegrated into fragments, and the cargoes were released during the process. Integrity of the cargoes is an essential step. So we validated that these nanoparticles were able to protect the siRNA cargoes. And since the objective or the therapeutic goal that we were trying to achieve was intestinal inflammation, so we were trying to administer these nanoparticles through oral route. So we made sure that even in the simulated or in the actual gastric and intestinal fluids, the cargoes were protected, and using this gel electrophoresis, we were able to verify that the cargoes were protected once they were within these nanoparticles. We measured the accumulation of these nanoparticles in an inflammatory bowel disease model. We developed the disease within these mice, and then we administer these nanoparticles through oral gavage. And as we can see that there was a varied distribution or accumulation of these nanocarriers within the intestines, and this was largely determined by the surface properties of these nanoparticles, with cationic nanoparticles accumulating heavily in the intestines, in the large intestine, compared with the carboxylated nanoparticle. And this was largely due to the presence of high excess of negatively charged mucin within the inflammation sites that was contributing towards a high accumulation of these nanoparticles within the large intestine. And there was some mucoadhesion as well as mucopenetration phenomena taking place within these different gastrointestinal compartments that were contributing towards the preferential accumulation of these aminated nanoparticles compared with the carboxylated nanoparticles. And finally, we tested these nanocarriers for inflammation therapy as well. We designed the therapeutic regimen with administering these nanoparticles over a certain period of 5 days. And then we tested at the end of the study, or during the process, we were able to determine the body weight, as well as at the conclusion of the time points, we were able to measure, take out the intestines and see how was the impact of the therapy of this RNA within an inflammation model on the therapeutic outcome. And we were able to see that our aminated nanoparticles, which were preferentially accumulated within those inflammation sites, were able to exhibit a high therapeutic performance compared with these carboxylated nanoparticles. We validated through histological evaluations, and as we can see that our aminated nanoparticles exhibit histological appearance of the tissues quite similar to our healthy models, but in case of carboxylated nanoparticles, they were less effective in preventing the inflammation of the intestines. So this study actually validated and provide a proof of concept of using these RNA drugs for the therapeutic application, and more specifically in an inflammation model. In the second part, I would like to share a story of visualizing these nanocarriers within the cells. For RNA delivery, we have to have the cargoes safely unloaded within the cells and able to interact either in the cytoplasm, or in case of a DNA drug, DNA cargoes, they need to be safely delivered to the nucleus. And one of the biggest compartment that any times the delivery vehicle encounters upon interacting with the cells are the lysosomes. These are the cell recycling centers, and they performed various useful recycling processes within the cells. But whenever we administer these delivery vehicles, they are also going to be taken up by these endosome lysosomes compartments, and either engulfed, destroyed, or able to escape. The cargoes are able to escape through these different compartments. And the traditional way of visualizing these lysosomes is through immune fluorescent staining, or by using some fluorescent probes that are weakly basic amines, or by using some reporter proteins that we can label them, and or sometimes using some enzymes, fluorescence enzyme substrates that have been employed to visualize these compartments. So in this second part, I would like to share a case study of lysosome labeling using a polymeric nanocarriers, and we were able to track the journey of this nanocarrier within the cells, and we were able to determine the utility as well as the efficiency of these various labeling methodologies in terms of the gene delivery. So we formulated these polyplexes using branched polyethylene, and we were able to formulate these polyplexes that were nearly 100 nanometers in sizes, and we were able to complex different RNAs, and we validated and characterized that these nanocarriers were robust and had a pretty consistent size through various batches. Our first goal was to see if there is any impact of these different labeling methods on the cellular health. We decided to use three different methods. We used a fluorescent probe, a weak basic amine, lysotracker red. We used the LAMP protein, lysosomal membrane protein, and that was either transiently expressed or a stably expressed version of cell lines. And we tested these different labeling methods in two different cell lines, the U87 glioblastoma cells or the breast cancer 231 cells. And we were able to see that that there was a slight decrease in the cellular health in case of the lysotracker red or the transient GFP, but in case of a stably GFP or the LAMP protein that was being expressed, we were able to see that there was an increase in the metabolic activity in 231 and U87 cells. We were able to see the impact of labeling on the cellular trafficking, and we were able to see that upon loading or upon delivering these polyplexes within these cells, we were able to see an enhanced uptake of these systems within the cells. A major therapeutic experiment was on the intracellular trafficking, and we were able to see these polyplexes within the cells using U87 as well as 231 cells, and we were able to quantify how much of these polyplexes are taken up by the cells. We can see that we have a polyplex signal within the cells, and we were comparing a lysotracker red as well as a stable GFP labeling methods, and we were able to see that in case of the lysotracker red and the stable GFP, we found a slightly different co-localization. These Mender's correlation coefficient, it is a quantified method of determining how much of endosome as well as the polyplexes are overlapping. That indicates that the polyplexes are trapped within these endosomes. When there is a less correlation or less overlap, that indicates that the nanocarriers or the polyplexes have successfully escaped the system. And we can see that in case of the U87, we had a high overlap in case of the stable GFP systems, which indicates that a lot of these polyplexes are entrapped within the endosomes. In 231 cells, we see less a slightly lesser or a significantly lesser overlap. That indicates that these polyplexes were able to escape through these in these cell lines. And this was directly correlated when it comes to the gene silencing efficiency. We tested these polyplexes in cells, and we were able to see that in U87 cells, we weren't able to see a very big difference between the gene silencing efficiency. But in case of 231 cells, we were able to see that that the stable GFP were able to show a much higher gene silencing efficiency, and this has largely to do do with the endosomal escape potential of the polyplexes in these systems. So in conclusion, I have tried to share with you two examples. One example of polymeric nanocarrier for delivery in case of an inflammation model, and we have shown that these systems are quite effective in treating therapeutic or in achieving therapeutic goals. And in the second example, I compared various labeling methodologies that we can use to track these nanocarriers within the cells. I appreciate all your time and attention, and happy to answer any questions that you might have. Thank you.